Good evening, ma'am.
Good evening, sir.
Nice. Thank you, ma'am. So good evening, my dear students. Once again, you all are welcome to the wonderful session of the biological class. Today, we are going to discuss about the part of microbes in human welfare, particularly microbes in industrial product. What are the microorganisms were used in industrial aspect? What are the beneficial effect is there? Okay. Before that, already we know very about last class. What are the subgetting? Already we saw it, isn't it, my dear? You know about what is by cheese, curd, and also yogurt. Okay. And how many types of cheese also present in the worldwide, how they are preparing, all those things already we discussed, isn't it, my dear? Okay. So these three things is very, very important. Cheese are classified into three types already in our about. Camembert, cheese of soft cheese, which microorganism, how many percentage of water molecule present in it, all those things already we discussed. Okay, my dear. And curd also. And curd also is prepared by the action of the lactobacillus, simply called as lab. Lactobacillus is also otherwise called as lactic acid bacteria. Okay. The starter culture is nothing but the inoculum. It contains millions of lactic acid producing bacteria. Okay. What is the main functions of the lactobacillus on milk? Okay, you know about the digest the milk protein, already we discussed, and enhance the amount of vitamin B12. It's also very, very important function. So many times they were asked the question in this part, my dear. And yogurt is also there. Yogurt is also one of the byproduct of the dairy industries. What about the flavor of the yogurt? You know about the lactic acid and also acetaldehyde flavor. Okay, which microorganism involved in this process? Streptococcus and also lactobacillus. Sour cream, streptococcus and Leconostac. Okay, bread also. Bread particularly, you know about the which one is used as a, which one you know? Bakery yeast. Bakery is also a common name, isn't it? The zoological name is Saccharomyces cerevisiae. Okay, different types of Saccharomyces is also there. Okay. What is by leavening? What is the meaning of leavening? When you are kept under the particular temperature, the flour must be, what happened? Swell up. That condition is called as leavening by the action of the enzymes produced by the yeast. They are all amylase, maltase, and also thymase. Okay. Why the flows are puffed in nature? How it will be soft in nature due to the action of the enzymes, not only that, and also the releasing of carbon dioxide during that, what is that? Bread making process. That things also already we discussed. Toddy. Okay, how many percentage of alcohol present in it? That things also already we discussed in this chapter. Other foodstuffs also. Now we are going to discuss about the microbes in industrial product. Particularly, there are so many microorganisms involved in this process. There are so many microorganisms used for the industrial product and commercial oriented, particularly fermentative beverages. Fermentative beverages, particularly tell us the alcohol, milk industry, dairy industries, cheese making industries, antibiotic productions, and ethyl alcohol. Okay. Even liquor factories, wine, gin. Okay. That things also we are going to discuss. See here, the fermentative activity of the microbes is used industrially to obtain a number of products. The two common one are alcohol fermentation and antibiotics. Particularly two things is very, very important. The microorganism most it used as a broad spectrum. Broad spectrum, what is the meaning of broad spectrum? It's mostly they are used in large scale. That is very, very important. Antibiotics and alcohol fermentation. What are the next one? How will the alcohol fermentation process occurs? You know about that. One of the important raw material, sugar molasses and beetroot molasses is also very, very important. And Saccharomyces cerevisiae, it's nothing but the brewer's yeast. Note it down, my dear student. This is very, very important. Zoological name of yeast is also, it's also one of the fungi, unicellular fungi. What does that mean? Unicellular fungi. Okay, fungi. This only unicellular, others are multicellular in nature. The zoological name of the, what is that? Saccharomyces cerevisiae. So this is a fungi. So botanical name you have to tell. Suppose Saccharomyces cerevisiae is a correct one. So mostly they were used for the production of beverages like wine, beer, whiskey, brandy, and rum, or by fermenting malted cereals only. They are isolated from the, which one you know? Malted cereals and fruit juice, and also the to produce the ethanol. Ethyl alcohol is one of the important substances isolated in the process called as distillation process. Okay. How it will be obtained? You know that malted cereals, fruit juice undergoes the particular temperature. Addition with the Saccharomyces cerevisiae, what happened? The fermentation process will also occurs and kept under the particular temperature for a particular period. After that, what happened? Already you know there are so many substrates that the substrates also digested, converted. Physiological changes occur with the help of the enzyme secreted by the microorganisms. As a result, what happened? You know, there is a product is that. That means that when the substrate combined with the molecules of the enzyme, what happened? You know, already you know about the traditional equation enzyme substrate complex is that. Then finally, what happened? Enzymes also release substrate converted into product. Okay. This product is a ethanol. Okay, my dear. Substrate, what is that? You know, sugar molasses, cereal juice, or whatever it may be, sugar. Plus enzyme, already you know, but enzyme secreted by the Saccharomyces cerevisiae. Suppose sugar contains sucrose, is a sucrose, isn't it? Plus is when you are adding the sucrose molecules, you are adding the enzyme, means nothing but the Saccharomyces cerevisiae. It's one of the fungi, isn't it? When you're adding this one, what happened? You know, automatically the yeast to secrete the enzyme is called as invertase. Don't forget, my dear student, invertase. Okay, and also sucrase. AAC is a suffix for all the enzymes already in our report. This enzyme act upon the sucrose, the disaccharide converted into glucose and fructose. Okay, media. So just to clarify it, this is very, very important. It's one of the most important equation is also there. Okay, media. Just to imagine. So sugar. Okay, sugar, molasses. Okay, sugar, molasses, what does that mean? No, it's nothing but the yeast. The yeast is a inoculum or fungi, isn't it? When you're adding into the sucrose, okay, the fungi secrete the enzyme. The fungi secrete the enzyme is called as what is that? Invertase. invertase, and also one more is that is called a sucrase. Okay, these are the two enzymes secreted by the yeast. So as a result, what happened? They act upon the substrate. They act upon the substrate. As a result, the sucrose converted into, because it is a disaccharide. Okay, disaccharide converted into two monosaccharide. Already you know about one is called as glucose. Okay, and another one is called as fructose. Okay, my dear, glucose and fructose. So after that, the glucose also undergo further a degradation process. As a result, sucrose converted into ethyl alcohol plus carbon dioxide. These are the important things, my dear. Okay, ethyl alcohol plus carbon dioxide. This is the traditional equations. Traditional equation they were followed. Don't forget, my dear students. This is very, very important. Okay, media. So this is very, very important. So go to the next one. Okay. See that the production of the different types of alcoholic drinks depends on type of raw material. Raw material may be vary for the fermentation and type of processing. This is very, very important, my dear students. There are so many alcoholic beverages also produced in the industries. So wine, take an example, 9 to 12% of the alcohol, and also the beer, 3.6% of the alcohol are filtered, pasteurized, and bottled without further distillation. How it will be obtained in the process called as distillation process. Note it down, my dear student. This is also very, very important. Saccharomyces is all nothing but the microorganism were used. Okay, so don't forget, my dear. These are the important things. Whereas distillation of the fermented broth is carried out for producing hot liquid, just whiskey, brandy, and rum. Okay. These are the important process. These are all our fermented process, byproduct obtained from the where industrial aspects. Okay. So go to the next one is this is consists of industry. See, there are so many containers there. Each one it has a specific temperature maintenance also there. Okay. Industrial aspect in large scale industries. Take an example in Tamil Nadu, Dasmark is one of the example. So what are the yeast? How many types of yeast have been used? Each one it has a specific properties and flavor, and also there. I have to note it down. There are 4 types of species. Yeast species also used for the alcoholic fermentation. First of all, you should note it down. These are the 4 types only. First one is called as Saccharomyces cerevisiae. It is simply called as brewer's yeast. Don't forget, my dear student. So many times they ask the questions. Okay. It is also obtained from the, which one sugar molasses. Sugar molasses. Okay, my dear. Don't forget. Sugar molasses. And second one is Saccharomyces ellipsoideus. It is also called as wine yeast. What is that? Brewery yeast and wine yeast. Okay. Wine preparation, they were used different types of wine preparation. And Saccharomyces sake. Okay, don't forget, my dear student. In Japanese people, Chinese people, it is also one of the favorite foods. They were used sake yeast. It is also so many times they were asked the question in 8 point, my dear. Soggy, just like which one toddy. Okay, it is also obtained from the one of the plant substance. And the last one is called a Saccharomyces pyriformis. Okay, it is also called a ginger beer or ale yeast. See the name, ginger beer, and also ale yeast. These are the 4 a type of yeast also used for the alcohol fermentation process by the industries. Okay, media. Go to the next one. See that this is the structure of the yeast. This is the structure of the yeast. Already you know, but yeast is the, first of all, you should note it down. Yeast is the one of the unicellular fungi. Okay. And also the fungi cell wall is made up of which one, you know? Chitin. Okay. Saccharomyces cerevisiaceae. The reproduction is also known as, but the budding. Okay. Reproduction also takes place. One of the asexual reproduction is called as, what is that? Budding process. That means, what is that meaning? You know, so take an example. This is consists a yeast. They produce a outer layer. There is a small bud-like structure is there. This is the mature yeast. Okay, this is considered a parent. Okay, the parent yeast. During the favorable condition, what happened? It also produced the protrude-like structure. Say, for example, this is considered a yeast, mean it produces a small bud-like structure, otherwise called as protrude-like structure, and peripherally. After that, what happened? It also undergoes the, what is the maturation stage. When it attains the maturation, automatically it will be this. What is happening now? Withdraw from the parent, and also it's also you will get it the new one. Again, it also undergo the further process. Likewise, the multiplication also takes place. It secretes the enzyme. There are two enzymes that already we discussed. One is called as invertase. Okay, invertase, zymase. Okay, the invertase is also called as, what is that? You know, sucrose. Okay, sucrase is the enzyme. Sucrase. Don't forget, my dear, this is the structure of yeast. Don't forget. When you are going to see the electron microscope, we can see the structure look like this one. Okay, go to the next one. See that the budding is also takes place in this part. See here, this is also two. Both are considered as a budding. See here, the initial stage. See, this one is called as budding stage. The protrude-like outgrowth is also originated from the parent cell. So once it attains this stage, what happens? It detaches from the parent, and also further it also enter in the other cycles, other life cycles, other asexual life cycles. Okay, media. Don't forget. And next one is, what are the nutritive medium that were used for the alcohol fermentation industries? That is also sometimes they may ask the questions. For example, barley meat for beer. So this is very, very important. So many times they were asked the question, which nutritive medium used for the preparation of beer? And fermented barley malt for gin. Same thing is there. Here the barley meat, here also barley malt. Fermented rice for sagi. Mostly they were used in the Japanese people, Japan, and also China. Don't forget, my dear student, it is also one of the NEET questions. Sagi is also obtained from the which means rice. Okay. And cashew apple for fenny is one of the, what is that? These are the product. Okay. These are the gin, beer, sagi, fenny, and potato ray for vodka. Okay, it's one of the favorite for many drinkers. And the next one is fermented cereals, barley, ray, maize, and also potatoes. Molasses for whiskey. Okay. Fermented molasses for rum. Okay. Fermented juice of the apple, beets, and also cherry for wines and brandy. So these are all of the processes. Why we are studying this one? No need to hesitate to study this one, because here you must note it on which nutritive medium they were used for the preparation of. These are the product. First of all, first you should note it down. Product. Okay. And second, you should note it down. Which one which nutritive medium is Which nutritive medium used specifically? That is very, very important. And third one is where it is produced. Where it is produced. All those things very, very important, my dear students. And one more is, sir, how many percentage of the alcohol is also present? Alcohol percentage is also very, very important in this part, my dear students. Sometimes they may ask the question, how many percentage present in the, how much of alcohol present in the brandy, whiskey, like that. They ask the question. Sometimes it's rarely, but note it down. Wine and beer are produced without distillations, whereas the whiskey 50% of alcohol. Already I told you. Now only I told you the whiskey contains 50% of alcohol. What is the meaning of 50% of alcohol? 50% of alcohol plus 50% of the water. What about brandy? It contains 70%. Okay, 30% remaining parts. Rum, it is also 40%. Gin is also 40%. Highest value, which one, you know? Brandy. And gin also 40% of alcohol produced by the distillation of fermented broth. This is the process, my dear students. How will you obtain from the industry? You know, one of the process is called as distillations. Just you may get it a drop by drop. Okay, that process is called as distillation. Fermented broth, the broth already in our nutritive medium. What are the nutritive medium that were used for the production of the industries? You know, the barley malt and also what is the rice and also cashew, apple, potato, fermented cereals, molasses, and fermented molasses and juice of apple, beech, and cherry. These are the broth. Okay, these are the broth. This broth also undergoes the fermentation process with the help of the microorganism. Is also called as substrate. The broth also called as which one, you know? Substrate. We are adding the microbes, isn't it? The microbes. The microbes utilize the nutritive substance from the substrate. They can produce the enzymes. The substrate converted into product, alcohol. It was isolated. It was separated by the process called as distillation. Okay, the number of traditional drinks and food are also made by fermentation by the microbes. Okay, so many things have been reported here, my dear students. See here, the microbes are also used for fermented fish, soya bean, and bamboo shoots to make foods. In Chinese people, they are used, which one, you know, the young part of the bamboo, young part. Just imagine this is considered a bamboo. Okay. So that is the tip, the meristematic. Young one is in this pot. Simply they cut and use it for the food. In industry, the microorganisms are cultured in a large fermenter. Just to note it down, this is very, very important. Large fermenter means it may be up to 10,000 gallons. Okay, gallons. That is very, very important. 1 lakh. Okay. The main functions of the fermentation is to provide a controlled environment for growth of the desired species of microorganisms to obtain a desired product. Okay. Here, 3 things is very, very important. What does that mean now? If you want to produce the fermentation product in good quality, desired quality means you have to select the broth. Isn't it, my dear? Broth is nothing but the nutritive medium. Suppose you are selecting the medium. So just imagine here a grape. Okay. So you will get it the, which one, you know, wine. Okay. Barley. You will get, what is that? Beer. Okay, like that. Because this is the medium. And what is a microorganism? There is a specific microorganism. Okay. Different types of microorganism, different types of substrate, and you will get the desired product. Entire, all the industrial product one of the main aim is what is that, ethyl alcohol production. Okay, my dear. See that how it will be occur, the fermentation process, how it will be happen, how the fermentation process occurs in the industrial aspects. Okay, there are two types of culture have been reported. One is called as batch process. Another one is called as what is that, continuous process. These are the two methods they were used for the fermentation process in the industries. First one is called as what is that, batch process. In this process, a large volume of nutrient medium. Okay, so nutrient medium with the volume as large as 2,25,000 liters. Imagine, already told you, I told you 1 lakhs like that, isn't it? But here they told 2,25,000 liters. It's inoculated and allowed. Inoculated means what is that? Microorganism also added to the substrate to proceed until maximum yield has been attained. Here, how much is the 2,25,000 liters? Okay, this is the inoculum, isn't it? When you're adding the inoculum with the help of the microorganism, when you're adding the microorganism, that inoculum or nutritive medium is produce how much amount? It may be 100-fold or 1,000-fold. Okay, folded than that of the which medium? The nutritive medium. So, for example, 2,25,000 liter is the inoculum, isn't it? The product is also multiplied into 100-fold, means just you add and what is that? 20. Okay, 30, 40, like that you add how much liter it will be obtained. Okay, so to proceed until the maximum yield is what is that, this much only. So just you add here 3 or 40. For example, 2,25,000 is the inoculum, and multiply into 1,000 or 10,000. Okay, you just add it here. Okay, so 1, 2, 3, 4 like that. This much of product we will obtain within the short period. Okay, madam, that is the maximum yield. That's why I told you 100-fold, 1,000-fold, and 1 lakh-fold. You will get it. That culture is called as batch process. Only one time only. So another important method is called as continuous process. The first medium is added either continuously or intermediately, combined by corresponding continuous or intermediate withdrawal of the portion of the medium with the fermented product. I tell you how it will be happen. Continuous medium. For example, this is considered as a fermenter. Okay, this is considered a large fermenter. Okay, mate. In this column, how much you are getting? There are two things there. One is inlet, input. You just imagine input, and also the output. Okay, so the output is also connected with the another tank. Here again, the process is also going on like that. Imagine. Okay, here in this gallon, what happened? First of all, you should note it down. Just for an example, I tell you, 20,000 gallons of broth, 20,000 liters, okay, of broth. Imagine here, okay, up to this level. Okay, my dear. So these are the broth Take an example, rice or wheat or apple juice or whatever it may be. You can imagine your own method. You're adding the microorganism. Okay. So the specific microorganism is there. Okay. Here what happened, the physiological changes occur. That means the conversion, the production of the enzyme is also occur, and also the enzyme converted into substrate into product. That process also occur. After that, what happening? It may be distilled. Distillation process. Distillation process occur. Okay. Distillation process occur means what happened? 20,000 liters of the broth. It gives how much, just imagine. 18,000 of liters of, what is that? Product is in first time. Again, you are adding, plus again you are adding the 20,000 of, what is that? Broth. And also addition of microorganisms. The same container. You are adding and adding again and again and again and again. It may be up to 10 times. 10 times. Okay. So how much it will be obtained? 18. Okay. You will get it some other product. This is the first product. Again, also you will get the 20. Again, 25. Again, 30. Like that. Okay. The process also increases. That we will not change the which one, the container and also and so, because once we were prepared aseptically already, about all the equipments or sterilized condition only, so the product is also continuously used, okay, for the production of the desired product. Okay, media, you got it, that point, I think. Okay, what are the next one? See that? That is the important things. And we will pass on to the next one, is very, very important, my dear student. Biotransformation of the steroids. Some of them intestines, what happened to the steroid components also secreted by the actions of the microorganisms, particularly, steroid and organic molecules. Steroid is also one of the, what is that, fatty acids derivative, fat. Okay, take an examples, fatty acids derivative, and also cholesterol. Okay, these are the content. And organic molecules, steroids are named because they are related to sterol that are abundant in nature. Example, cholesterol and stigmasterol, ergosterol. See here, the cholesterol is nothing but one of the fat molecules, semi-solid molecule, is mostly found in the human body, where brain and nerve tissues already where is the deposition takes place in your body, just imagine so many places there, particularly adipose tissues. What is that? Adipose tissues. The fat deposition takes place in your skin is called as, what is the main important points, my dear, adipose tissues. Other than the adipose tissues means you have to take an examples, which one, brain and nerves and also kidney. Okay, and the last one is called as, what is the stomach. Okay, that's why we are getting the belly. Okay, that is very, very important. So the cholesterol found in brain and nerve tissues, and stigmasterol, stigma already one of the female part of the flower, so it is also present in the vegetable soils. Ergosterol, ergos is nothing but the fungus. Okay, my dear, fungus. So the ergosterol in yeast, sapogenins, diogenins, and hecogenin in plants. Sapogenins, diogenins, hecogenins. These are the things. Sometimes they may ask the question, which one of the following is a ergosterol? Okay. And cholesterol, where the deposition takes place in your body, a human body, other than the adipose tissues, like that they may ask the question, find your students. That is very, very important. Nowadays, there are several, what is that? Microorganisms. Take an example, several microorganisms also. What is that? Several microorganisms. Okay. Such as Rhizobus, nigerians, Curvularia, lunata, Aspergillus, already know about penicillin, Gliocalidum, Fusarium solani, and some, what is that? Yeast and fungi are used for steroid biotransformation. Okay, my dear. So these are the microorganisms mostly used for the production of which one, a steroid biotransformation process in the large scale industries. That is very, very important, my dear. Particularly sapogenin is one of the examples they have given. The sapogenins are extremely useful starting material for the synthesis of sex hormones and also for the synthesis of corticoid and contraceptive drugs. Very, very important. Already know one of the, which one starting material, sapogenins. Sapogenins is also isolated from the ergot fungi. Okay, fungus. What is the main use of this one? The sapogenins for the production of the sex hormones. Already there are so many sex hormones there, and male as well as the female is also respectively, isn't it? Male hormones are androgen and testosterone, female hormone progesterone, oxytocin. Okay, luteinizing hormone. So many hormones is there, male as well as the female. They are having a separate sexual hormone is the female. Okay, my dear. Here, very, very important for this one, corticoid, mineralocorticoid, non-mineralocorticoid. There are two types of that, corticoid. And contraceptive drugs is mostly used for the controlling of the control effect. So what are the next one is called as antibiotics. Okay, my dear, this is very, very important, my dear students. First of all, beverages already we discussed. Second important point is the interstitial product of the antibiotics. Very, very important, most important in this question. So many questions asked in this part only. Maximum 4 to 5 questions maximum in this part. Maximum means this lesson I am telling. The antibiotics are chemical substances secreted by some microorganism. Antibiotics are chemical substances. That chemical, it may be a protein. Okay. And also lipid. And also alkaloids. Okay, alkaloids. Alkaloids. Okay, my dears, this is very, very important. So that antibiotics are chemical substances secreted by some microorganisms that inhibit the growth and development of the other microorganisms. One microorganism secretes the compound that will be affect the another microorganism. Such type of component is called as antibiotics. For example, you know about the rabies. Rabies is one of the diseases, isn't it? One of the diseases obtained by the biting of which one, dog. Dog salivary gland, it contains the rabies virus. Is so much is there. Okay, dog biting. How will you get the remedy? An antibiotic means you will get the antibiotic from the same microorganism, say rabies. The rabies microorganism is also isolated from the salivary gland of the dog, and also it can be attenuated or some other physiological changes which occurs within the laboratory and also modified into what is that? Some components, modified component. It inhibit the which one, exact the same microorganism. One more is that COVID, isn't it? COVID. Already we discussed. COVID is the one of the worldwide problem created 2 to 3 years. Okay. We are using that, which one, injection or antibiotics, isn't it? Vaccination. The vaccination contain what is that microorganism idea? Same COVID microorganism present inside it. Whether portion of the COVID virus, whether it may be a protein part, whether it may be nucleic acid, whether it may be a that's hooks, the structures. Okay, just similar to that of the HIV virus. There is a protein molecule there, that is hooks, isn't it? Glycoprotein, isn't it, my dear? Inside there is a single standard, which one, RNA or DNA. Here the RNA molecule is there. Okay, RNA molecule covered by the so many glycoproteins. Lipoprotein also, the outer membrane also present. And inside there is a reverse transcriptase enzyme is also present in it. Isn't it, my dear? Okay, how it will be controlled? How will we prepare the antibiotics against this microorganism? Means you have to select the microorganism. You have to collect the any part of the body. This is called as glycoprotein, GP. Here inside there is a nucleic acid. You have to cut and isolate from the nucleic acid from this particular viral DNA or RNA molecules. After that, it may be modified in the laboratory aspect. Okay, after the completion of the project or process, it may be tested so many times. It may be tested so many times. After that, it may be entered into the marketing places and also distributed to the peoples. Most of them are produced by the actinomyces. Don't forget, my dear students, maximum antibiotics production takes place with the help of the fungi. It's called as what is it? Actinomyces, mycology. Especially the genus Streptomyces. Streptomyces is nothing but the, what is that? Bacteria. Okay, streptomycin. Already you know, but those who are not accepted their body by the penicillin action, the penicillin antibiotics, they will not accept. Such time what they will provide, the alternative form of the penicillin, they will provide streptomycin. Okay, here the two things is that actinomyces is one of the filamentous fungi. Don't forget, my dear student, actinomyces. Okay, streptomyces is nothing but the bacteria. Okay, these are the two important things, ma'am. The ancient ages, first of all, you should note it down. The study of antibodies began by the discovery of penicillin. The penicillin is the starting one. So first discovered, which one, antibiotics in 1929, 28, actually 28, 29. When Alexander Fleming provided that the filtrate of broth culture Penicillium notatum, don't forget, my dear student, has antibacterial properties in relation to the gram-positive bacteria. Here you must note it down. 3 points is very, very important. No, not only that 3, there are 5 points is very, very important. Most of the antibiotics produced by which type of microorganisms? Actinomyces is nothing but a fungi. Take an example. Actinomyces is a fungi. And which bacteria they ask, sometime. Okay, you have to answer it. Which one, Streptomyces. Streptomyces, it's streptomycin. Don't forget. Okay, my dear. And then second question is, what is that, the penicillin discovered year? Okay, 1929. Okay. Who discovered? Alexander Fleming, already you know very well, but even in 5th standard answer and we are studying these things. Alexander Fleming. And which fungi they were used as a production of penicillin? Okay. Culture broth, the Penicillium notatum. Don't forget, my dear students. So many times they were asked the question in this part. Penicillium notatum has antibacterial properties in relation to gram-positive bacteria and gram-negative, isn't it, my dear? But in this case, the penicillium act upon against the gram-positive. How will you differentiate gram-positive gram-negative bacteria? You know well about that. It also depends upon the a cell component. NAM and NAG. What is the meaning of NAM and what is meant by NAG? N-acetyl muramic acid and N-acetyl glycosamine, otherwise called as glycocalyx. Okay. The content is also vary. Okay, these are the very, very important points. So here you must note it down. Antibiotics is nothing but a chemical substances secreted by some microorganism that inhibit the growth and development of the other microorganisms. Most of them are produced by the actinomyces fungi, especially the genus Streptomyces and the filamentous fungi. Streptomyces is nothing but the bacteria. The study of antibiotics begins by the discovery of the penicillin in 1929, when Alexander Fleming provided the filter of a broth culture of Penicillium notatum. It has antibacterial properties in relation to the gram-positive bacteria. However, its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Fleury. After that, what happened now? The effective antibiotics, Ernest Chain and Howard Fleury. What are they? First of all, is noted this antibiotic was extensively used to treat American soldier wounded in Second World War. Sometimes they may ask the question, Second World War, what happened? Who discovered the effective antibiotics like that? Okay, so this is very, very much. Take it out and take notes, hints also. The Fleming, Chains, and Fleury were awarded the Nobel Prize in 1945. This is also very, very important. When they were get Nobel Prize for their discovery. 1945, 1929. Difference, how much is that? How many? 16 years. After 16 years, he got the Nobel Prize. The Second World War occurs in 1945 only. Okay, started. Just that thing, that time they got Nobel Prize. Okay, made it. Go to the next one is, what is by broad spectrum antibiotics? The broad spectrum is nothing but the single antibiotics fight against many diseases. Many diseases. Such type of antibiotics are called as what is the broad spectrum antibiotics. Are effective against a broad range of microorganisms. That is nothing but a single what is that antibiotic. They may control 50. They may control the 50 microorganisms. For example, I am telling, but exactly I don't know. Some microorganisms, diseases. That is called as broad spectrum antibiotics. Okay. And bactericidal means, what is that meaning? That some of the microorganisms, what is that? Produce the antibiotic that kill the bacteria is called as bactericidal antibiotics. Antibiotics kill the bacteria, whereas the bacteriostatic antibiotics halt growth of the bacteria. What is the difference between these two? This is very, very important, my dear student. This is very important. Bactericidal means it kill the bacteria. Bactericidal means suicide, kill the bacteria. Some of the antibiotics, they may kill the bacteria, means they are called as what is the bactericidal properties. Bacteriostatic, what is the meaning of bacteriostatic? Means, for example, it will be control the growth of the bacteria. So that is called as halt, means what is it? It will not grow. After that, it will not grow. Completely stopped. That is called as what is it? Bacteriostatic. Don't forget, my dear students, very, very important in this part. Okay. See the next one. Sir Alexander Fleming, they have given us a small introduction already in our board. So very, very important, my dear students. Who is Alexander Fleming? How he discovered which microorganisms used for the production of the antibiotics, all those things, which enzymes they were secreted, all those things you have to note it down. Sir Alexander Fleming, a Scottish biologist. discovered the enzyme lysozyme. Lysozyme is also one of the enzyme already in our board. Lysosome mostly present in the where lysosome secrete. How many enzymes there are so many enzymes, 60 enzymes also secreted by the single cell bound organelle. This was first introduced by the name of the scientist called as, what is that? Alexander Fleming. Don't forget, my dear, not only the antibiotics, penicillin, and also very, very important, before the discovery, the penicillin also discovered 1929. Before 8 years, so 1921, he discovered first, what is that? Lysozyme. Lysosome is nothing but a suicidal bag, already in our board, isn't it, my dear students? It can be active upon the phagocytosis process. Once what happened, the foreign particle inverted into the microorganism, any other courses, automatically what happened, the lysosome act upon it. Okay, it's secreted so many microorganisms, enzymes. The enzymes act upon a molecules, complex molecule, completely digest and gives a simple molecules. That's why it is called as, what is that? Suicidal bag, lysosome. The lysosome secrete the enzyme is called as lysozyme. Okay. The lysosome, what happened, it act upon the bacteria cell wall and lysis. That's why the process is called as lysozyme. And the antibiotic penicillin 1928 from the fungus Penicillium notatum while working of the staphylococci bacteria. He preferred, this is also very, very important. When he discovered the Penicillium notatum while working staphylococci bacteria. Don't forget, my dear students. During this process, he discovered this one. Because the penicillium look like a structure is also similar to the staphylococci. Because penicillium already in our one of the fungi, isn't it, my dear? The structure will be like this. Okay. The structure will be the penicillium. The structure will be like this. This is called as gonadiophore. This is called as gonadiophore. Already you know about penicillium structures. You just saw it. This is called as gonadiophore. Just to note it down, my dear student. This is also very, very important. Gonadiophore. Gonadiophore. Don't forget, my dear students. So the structure is look like which one, staphylococci. Staphylococci structure. Cocci is nothing but the round shape. The structure of the bacteria is also look like this one. Okay, the structure is called as staphylococci. Streptococci look like what is that? Stay one above the others like this. It's just nothing but the bunch of, what is that? Egg. Okay, this is called as, what is that? Streptococci. Strepto. This one is called a staphylo because the structure is similar to that of the yeast gonadiophore. That is very, very important. He prepared the treatment for the bacterial infections such as syphilis, gangrene, and tuberculosis. Okay, this is very, very important, my dear. Lysozyme exists in tissues and secretion like a mucus, tears, and egg. So egg white. Let's note it down, my dear students. Lysozyme, where it is it? Just to note it down, mucous membrane. Where is the mucous membrane? Is that just take an examples of nose? There is a mucous membrane is there. Small intestine, urinary tract, all those things. It is also made up of which one in a mucous membrane, which contain lysozyme enzyme. What happens? Automatically, you are having in the natural immunity powers. How it will be present in because the presence of lysozyme, it act upon it. So as a result, what happened? The bacteria completely elucidated, digested, killed. Okay, and another one is called tears. Tears means lacrimal gland. Lacrimal gland. Just imagine your eyes also containing which one eye also containing the lacrimal gland. In this part is called as lacrimal gland. Okay, in this part, what is that? In this part, what is that? There is a what is that? Okay, okay, this part. So this is called as lacrimal gland. The lacrimal gland secrete the which one tears. Tears contains antibacterial properties. Okay, so not only the tears, and also one more is that egg white is also, it has the antibacterial property. Don't forget, my dear students, it's contained. Here, what they are telling, lysozyme present in various mucus, tears, and also egg white. Lysosome, mucus means what are the organs which present in mucus, means small intestine, urinary tract, nasal secretion, nose. Okay, tears, lacrimal glands, and also the tear also containing what is it, lysozyme. Egg white is also containing which one, lysozyme. So these are the important, my dear student. These are all are discovered by the Alexander Fleming. See the example. How did they make the penicillin? So this is considered a penicillin, isn't it, my dear? Penicillium notatum. Penicillium mold naturally produce the antibiotic penicillin. Already you know about microscopic view of the penicillin. The penicillium growth under the broth container, fermented tank. The scientists learned to grow the penicillium mold in deep fermented tank by adding kinds of sugar and other ingredients. Kinds of sugar is nothing but the DTT medium. The process increased the growth of penicillium. So here inside what happened? The multiplication there, the fungi absorb the nutrition from the tank and also it undergoes the multiplication process. As a result, the counting will be increased. So this is the penicillium molecules, how it will be obtained like this. Then scientists separate the penicillin product from the mold. The mold from this one, what happened, penicillin is also isolated. It's also commercially prepared. But in ancient stages, they collect the what is that? Penicillium product from the molds. After that, finally, penicillin is purified for use as an antibiotic medicines. This is normal one. Ancient ages, they were used like this. Nowadays, there are so many industries also. Okay, they are preparing penicillin. Instead of penicillin, they were used streptomycin, and that is very, very important, my dear students. Nowadays, the penicillin production is also stopped because it is not favor for all the human beings. Some of them are getting allergy. So that's why what happened, divide it. Okay, go to the next one. See, this is also another process, production of the penicillin. So slant culture, shaker flask, transfer vesicle, seed tank, fermented tank, and rotatory filter. After that, purification of the column, and second extract, third extract, vacuum, slurry, and ceramic filter, crystalliform, crystal vases, ceramic filter, and vacuum dryer, crystalline potassium, and penicillin is recovery. These are the process. These are the process occurs in the intestinal organ. Finally, we'll get the powder form, penicillin recovery. After that, it's also mixed with the distilled water and also injected into the patient. Okay, my dear. And some clinically important antibiotics have been reported in your book. Okay, here, very, very important, my dear students. First of all, is note it down how many? 13, I think. Okay, 13 microorganisms, antibiotics also produced in laboratory. Here, what are the parameters they have given? First of all, is it noted down serial number is there, and also antibiotics. How many antibiotics? 13 antibiotics is also there. Produce organism, which organism from the antibiotics also produced. The activity, whether gram positive, gram negative, broad spectrum, dermatographic, like that. The mode of action, how it will be occurs inside the physiological activities inside what happened. That thing is also very, very important, my dear students. First of all, the penicillin. Penicillin is also one of the important antibiotics, most old, ancient antibiotics produced by the Alexander Fleming in the year 1928. Already about. This is isolated from the microorganism is called as Penicillium chrysogenum. Okay, chrysogenum. Okay, it is also activity against the gram-positive bacteria. So what is the mode of action? Is cell wall synthesis. Inhibit the cell wall synthesis of the bacteria. Cell wall synthesis is very, very important. Suppose without cell wall synthesis, what happens? It will not undergo the further multiplication process. So the reproduction is very, very important for the mode of transmission. Okay, and also enhance the disease Disease. What is that? Characteristics, isn't it? It enhance the rate of reaction. That is very, very important. Cephalosporin. See here, that is also very, very cephalosporin acrimonium. Cephalosporin acrimonium. Don't forget, my dear, cephalosporin acrimonium. Here, chrysogenome. Okay, my dear, chryso. Okay, so gram positive. So broad spectrum. Broad spectrum means what is the meaning? No, already I told you. One, antibiotics, they act against many diseases, isn't it? They control many diseases. Just such type of antibiotics are called as what is that? Broad spectrum. It is also similar to that of cell wall synthesis. Fumiglin, Aspergillus fumigatus. There is no activity. Cell wall synthesis. Chrysoflavin, Penicillium chrysoflavin. Okay. Dermatophytic fungi. Okay, sorry. Dermatophytic. Dermato is nothing but a skin-oriented skin. Microtubule formation. And bactericin. Bacillus subtilis, gram-positive bacteria, cell wall synthesis. Polymyxin B. Bacillus polymixa, gram-positive bacteria, cell membrane. Here cell wall, here cell membrane. You know the difference between these two. Gram-positive bacteria, it contains cell wall as well as cell membrane. Don't forget, my dear students. Erythromycin, already we saw it, these things also. Streptomyces erythraeus, gram-positive bacteria. Protein biosynthesis inhibited by the protein biosynthesis, because protein biosynthesis is also essential for the production of the new organism of the. Okay. So the protein bias is completely inhibit, means the new organism it will not produce, they will not undergo the further development. So that's why they used which one now? Erythromycin. And also neomycin, that is very, very important. Streptomyces phrediae, broad spectrum protein biosynthesis inhibition. Okay. And also Streptomyces crazius, gram-positive bacteria, protein biosynthesis. Okay. And also tetracycline, already know, but very, very important. Tetracycline, most of the cases, tetracycline, oxytetracycline or terramycin. Streptomyces erythraeus, very, very important, my dear students, NEET. And neomycin is also. Okay. It is a broad spectrum protein biosynthesis. Here they may ask the question in this part, very, very important. Which one of the microorganisms, they also act as a broad spectrum manner, and some of what is the main inhibition process, protein biosynthesis. Rifamycin, another one is called Streptomyces mediaterinae, tuberculosis, that is also very, very important. TP, protein biosynthesis. Chromopenicol, Terptomyces venezuelae, protein biosynthesis. There is no any action here, positive or negative or whatever it may be. Novobiaxin, Streptomyces nivis, and also Streptomyces spheroids, example, inhibit DNA polymerization. It is also different, isn't it, my dear? These are the important things, important of the what is that? Antibiotics, and also which organism is produced. That is also very, very important product of the organism, and also activity against the gram-positive or broad spectrum or dermatophytic fungi or some other things, whether bacteria, virus, or something else like that. And mode of action is very, very important. This column is very, very important, my dear students. Which animals is also very, very important. Which antibiotic, these things is very, very important, my dear students. Go through it and also study well, and you will get the good result. Okay, my dear. So we will see the remaining things on the next class, my dear students. We will finish it up to this level only. Okay. Thank you so much. Bye. Take care. Thank you, ma'am. Hello, ma'am. Thank you.
Thank you, sir.
Thank you.